to presentation, formulation of research strategies for standardization and value generation of substances derived from natural cannabis resources. Before I start the presentation, I would like to thank you to Professor Jin Jong-sik for their kind invitation, as well as to all the individuals associated with the ISAMS committee. I have joined the College of Pharmacy at Jeju National University, as an assistant professor of pharmaceutical science, starting from last year, 2022. Prior to my current position, I was working at the University of Alberta in Canada for 3 years from 2019 until August of 2022. Cannabis has gained significant attention in recent years due to its potential therapeutic benefit in various medical conditions. Here is today's content of the presentation. Our focus has been on standardization and value generation of natural cannabis resources. In the introduction section, I am going to talk about the brief outline of cannabis research about clinical evidence. And second part, the cannabinoid acid-loaded microimmersion. I'm going to introduce about the field of cannabinoid delivery and provide valuable insight into harness the therapeutic benefit of cannabinoid acid. Third section, standardization, value generation of substances. I'm going to talk about in terms of stream technology. And the last part, I'm going to provide the insight for whole cannabis-related researches. Looking at Canada's cannabis legalization process, their movement includes decriminalization of cannabis and legalization of medical cannabis and legalization of recreational cannabis. Regulations on cannabis have been gradually opened as a result of ongoing medical and scientific research for cannabis in Canada. Cannabis has side effects unlike other drugs. It has a high utility value, minimize side effects, and is very effective in treating necessary medical disease. Canada proposed legal guidelines to emphasize the need for medical cannabis through the risk-benefit analysis. Humans have been using whole flowers, fruit, and seeds for recreational, medical, and industrial purposes since 5000 BC, before Christ. More than 455 clinical trials are underway in North America alone, and approximately 53 in Canada, depending on 117 patient condition. 4 different cannabis-derived drug products were approved in 2019 and have been applied to various kinds of patients in South Korea. Extensive clinical trials are underway regarding the dependency, toxicity, and drug efficacy of medical cannabis. In most current cases of cannabis, the raw materials are most commonly used as a ready-to-use form of smoking. The route of administration through smoking is absorbed into the lung mucus, but this one causing the carcinogen and lung toxicity. And since the physiochemical properties of the cannabinoid that has hallucinogenic effect are very lipophilic, it is mainly manufactured in a form that can be taken by dissolving it in oil, or in a form that can be taken by encapsulating the oil formulation in soft form. The clinical usefulness of medical cannabis is very broad, as it can be applied to a variety of disease and symptoms depending on the level of evidence. According to the level of evidence, in cases where the clinical results support the pharmacological mechanism is very strong. The first conclusive or substantial evidence of efficacy showing the strong clinical evidence. Specifically, Epidiolex, a CBD drug that shows an excellent therapeutic effect for pediatric epilepsy, was added as an adjuvant treatment for tuberous sclerosis in April last year. In this slide, I'm going to briefly explain the classification of cannabis based on its chemical component. Cannabis, also known as marijuana, can be distinguished into various categories based on its chemical composition. And then you can see the table. The chemicals collectively termed cannabinoid can be organized into three broad classes or categories. The first one is phytocannabinoid. It is the plant-based, the individual molecular constituents of the cannabis plant. The second one is, the last one is the synthetic cannabinoid. It is a man-made cannabinoid. And the endocannabinoid is the produced by and within the body. Cannabis can be classified based on its chemical component as outlined in the following table. This classification system helps distinguish different types of cannabinoids based on their chemical makeup, including the types of cannabinoid present, the ratio of THC to CBD, or some of the terpene profile, flavonoid composition, and other chemical compounds found in the plant. Almost 1,600 different compounds found in the cannabis. Additionally, cannabis contains various flavonoids and other chemical compounds that also contribute to its overall chemical complexity. Cannabinoids are a diverse group of the chemical compounds found in the cannabis plant with over 100 different types identified so far, but nowadays they are founded, and then over 500 cannabinoids are almost constituents are included in the cannabis. And then most well-known cannabinoids include THC and CBD, each with distinct effects on the body and and then other types of cannabinoid, CBG or CBN, THCV, also gaining attention for their potential therapeutic properties and unique mechanism of action. In the body, there are cannabinoid receptors called CB1 and CB2 that interact with natural analgesic and anti-inflammatory substances. Both receptors are G-protein coupled receptors, with CB1 primarily expressed in the brain and minimally in peripheral tissues, while CB2 receptor is mainly found in the immune cells and hepatopoietic cell. In the past, some of the big pharma tried to develop the new drug based on this endocannabinoid system, but it showed very adverse effects, like for reducing patient body weight. So that's why they are failure to approve the new drugs. Cannabinoids have varying molecular weight ranging from around 300 to 400. They are very lipophilic and low water solubility, being more soluble in non-polar solvents like ethanol and oils and fat. And they are very sensitive to light and heat and oxidative conditions, requiring storage in a cool, dark, and dry environment for stability. Cannabinoids can also exhibit pH-dependent solubility and stability with potential interaction affected by changes in pH. The types of metabolites produced during cannabinoid metabolism and the pathway through which they are transported within the body vary depending on the stage of metabolism. As a result, the patterns of change in blood concentration can significantly differ. Some cannabinoids do not always follow this consistent pathway to their final metabolites, and certain metabolites can have psychoactive effects. This outline for the cannabinoid acid-loaded microimmersion research article. Additionally, we are getting more interest to achieve the ideal delivery efficiency of cannabinoid acid. And this slide, this study provides the structure for discussing the patient-centered approach utilizing cannabinoid acid in microimmersion formulation. This part, I'm going to talk about the rationale for this research. Through the utilization of the microimmersion system, this cannabinoid acts as the model drug, so it has possess potential therapeutic opportunities, but are very highly unstable and limited bioavailability. That's why we choose this cannabinoid acid as a model drug, and then try to improve the stability and then permeability in the in vitro transdermal system. The cannabinoid acid such as THCA and CBDA have shown promising therapeutic potential. The THCA has anti-inflammatory activities, and second one, neuroprotective and anti-neoplastic effect. And then CBDA also very similar therapeutic potential, and then it has anti-inflammatory activities and anti-emetic activities, and even it also has anti-tumor characteristic. In summary, while model cannabinoid acid hold promise for their therapeutic potential, their instability, limited availability, and ongoing research effort to fully elucidate their effect present challenges and limitations to their widespread use. So here is the flow chart for cannabinoid acid-loaded microimmersion. So first, we try to screen the oily vehicle and surfactant vehicle to which oily vehicle and surfactant is highly solubilization effect to the cannabinoid acid, and then we first try to set up the formulation composition of the microimmersion vehicle, and then design and evaluate the microimmersion. We add the model drug to the microimmersion vehicle and try to check the stability and particle size and zeta potential, and then try to see the instability of the microimmersion vehicle. And then third, we try to choose the highly stable formulation of the microimmersion, and then try to check the performance of the microimmersion. So we using the in vitro permeation study using the front diffusion cell system, and then in the fourth part, when we try to adjust the pH of the microimmersion, then we can significantly improve the permeation effect and stability of the microimmersion. Then finally, we got the highly stable microimmersion for 6 months. In this part, we check the oil and the surfactant to check the permeability and then solubility of each model drug, and then try to select these green boxes of the surfactant and then red box of the oily vehicle to make the stable microimmersion. So We also, before loading the drug, we optimize the composition of the vehicle, which serves as the base, using phase diagram and evaluate its stability. The model drug used in this study was the cannabinoid exit. So qualitatively, we also see the emulsification of ability of each microimmersion and try to check the compatibility assessment and formulation stability evaluation through the dilution. And then last part, stability evaluation based on drug incorporation. And then we select the 4 kinds of the main surfactant to make the these kinds of almost 40 kinds of formulation, and try to see the which region is the most stable microimmersion. So the gray region is the composition ratio to make this kind of ternary, pseudo-ternary phase diagram to make the compare when loading the drug, and then some of the surfactant significant change the region of the ternary phase diagram. So that's why we choose the C. Using the Proseti AWS, that non-ionic surfactant is very effective to maintain their gray region in the ternary phase diagram. So we also see some tendency when I control the composition of the surfactant. We can also see the tendency of the physiochemical property, and then they have a unique structure with tiny droplet of oil dispersed in water, and then even this kind of things also support the Caprio 90 and Procetyl AWS can maintain the under 200 nanometer of the particle size. So we also see, as you see on the slide, we can see the Proseti AWS composition demonstrate the distinct and uniform size distribution of microimmersion particle. So we choose these Proseti AWS as the main surfactant for the cannabinoid exit loaded microimmersion. So in vitro plant cell also show the significant improvement of the permeation effect. So, but we focused some of the microspecies of cannabinoid exit exhibit unique characteristic as function of pH. So if we control the pH, then we can also significantly improve that plane vehicle. So it also understanding the pH dependent behavior of the microspecies of model drug, it can also very critical in optimizing their pharmaceutical properties, including solubility, absorption, and targeted delivery. Then optimized pH of each formulation have different pH. So the red plot is the previous optimized thing, but we slightly control the pH, then we can also improve the permeable effect to the cannabinoid exit loaded microimmersion. Then we have investigated the stability profiles of the optimized product. Then we can also qualitatively and then quantitatively to evaluate the profiles of each cannabinoid exit. In this part, I'm going to talk about the field of cannabis industry. Understanding the flow of operation, often referred to as the stream. This stream can be broadly categorized into 3 main segments. The first one is upstream, and then second one is midstream, and the downstream. It is very important for academia and industry in the cannabis sector to focus on each field and explore foundational technologies. The cannabis industry can be divided into this kind of three mainstream. So the next slide. In the upstream part, the medical cannabis upstream technology patent data showed in the following areas. The first one is seed and crop protection and cultivation method and equipment, precise crop harvesting in situ, post-harvest method, and genetic engineering technology and disease and pest control is the kind of upstream part. So these topics are very focusing on the original cannabis, natural cannabis resources, and additionally, new strain trait and genetic manipulation and editing are advancing now, and which may affect the discourse around medical cannabis. In the midstream part, the major patenting activity in this part of the supply chain is still in natural product technology, namely extraction of cannabinoid, terpenes, and flavonoids, as well as purification, separation, and preservation. The challenge here is to provide easily scalable extraction method with the high yield of the desired compound, chiefly the non-psychotropic ingredient CBD. Other potentially medically useful compounds referred to as an entourage compound are present in low concentration in the natural state and require the development of meticulous extraction, separation, and purification technique. In the downstream part, medical cannabis extraction obtained from midstream technology are formed into composition, formulation, and compound for each, which patent protection is sought to in regard to the treatment of a number of medical conditions. Actually, the repeat-based cannabinoid delivery system utilize repeat as the primary component to encapsulate and deliver cannabinoid. Even the polymeric-based cannabinoid delivery system employ polymers as carriers for cannabinoid. This kind of delivery system offer advantages such as improved drug solubility, control release, and improved bioavailability. This is conclusion for the upstream part. The historical development of cannabis and CBD research can be clearly divided into this kind of three stages, which focus on first chemistry, pharmacology, and molecular biology aspects of cannabis sativa in general, and then focus on CBD-related publication. Still the upstream part, we should focus to control the quality and then safety management of cannabis contaminant. It's very important for the extraction of medical ingredients like potency, terpene, moisture content, pesticide, heavy metal, and microbiome. That kind of contaminant is also should be controlled and then have to establish the criteria. And the second part, we have to investigate the critical quality attributes of cannabis crop and establish the systematic SOP, ranging from cultivation environmental conditions that may affect it, post-harvest management, extraction, processing process, and storage condition. Gangwon Special Self-Governing Province and even Jeju, Gyeongsang-bukdo, and Jeollabuk-do attempt to standardize hemp cultivation based on smart agriculture. And then medical cannabis production using aerated plant factory technology also have been tried by Jeonbuk National University. So this kind of the systematic culturing system have to be more systematic approach. And then in a midstream part, we have to think about consider about there are several extraction methods, including ultrasonic disruption, extraporation, and organic solvent extraction. And this one is the last part. Medical cannabis extraction obtained from midstream technology are formed into composition, formulation, and compounds for which patent protection is sought in medical condition. Especially in the left part, the cannabis-derived drug product should be nowadays focused on the palliative care, and then in the oral formulation, going to be very low bioavailability, so they are moving toward personalized, safe, and effective solution like non-invasive delivery of the transdermal formulation. So, yeah, thank you for attention.